Yu-Ning Huang. I'm an incoming PhD student at USC School of Pharmacy. Yeah, it's glad to join an in-person conference to give a presentation. I love medicine. Yeah, so today I'm going to deliver the first topic that is also an ongoing project within our lab, which is titled as Assessing the Completeness of the Immunogenetic Database Across Diverse Populations. And yeah, slide just shows you a brief introduction about what's the basic immune responses. And the immune response will be triggered by when there's a pathogen or what we call the xenobiotics when enter the human body systems, and it will be engulfed by what we call the macrophage, and then macrophage will present its the antigens to itself, its surface of the antigen, its surface, and then will further trigger what we call the helper T cells. And then the helper T cell will then further trigger what we call the B cells and T cells, or B cells will turn into plasma cells, and then it will help secrete antibody to fight against the virus, the infected cells. And for the other pathway, that the helper T cell will further activate the cytotoxic T cells, and directly the cytotoxic T cell will kill the infected cells. And yeah, this is just a brief mechanisms of the immune responses. And when it's involved in B cells and T cells, that's what we know as the adaptive immune responses. And when we talk about the immune responses, the most important part of the adaptive immune responses are the BCR and TCR, which is a B cell receptor and a T cell receptors. And the figure A just shows that the B cell receptor and T cell receptor are really similarly organized. Like B cells are composed of heavy chain and light chain, and the T cells are composed of alpha chain and beta chain mostly. And the most important part of these BCR and TCR are the tip of the part, which is the variable region there, which there's an antigen binding sites. And these antigen binding sites are encoded by what the VDJ recombinations. So the figure B that we just want to show is the recombination of the VDJ genes here. Yeah. And the motivation of our study is because this is a previously published research in our lab that is published in Cell Immunity that we just shows that the proportion of ancestry in the TCR using studies are mostly composed of European ancestries. And yeah, the average number of the published, the average number of the European ancestries also have a larger number compared to Asian and African ancestry from the figure in the right. And so our goal in this project is just trying to examine how's the completeness of the immunogenetic database that is representing diverse populations. And the immunogenetic database that we're referring to is the IMGT database, which is the International Immunogenetics Information System database. And this is a database that is majorly comprised of immunoglobulins, T cell receptors, and also the major histocompatibility of human and other vertebrate species. Yeah, just to give a brief introduction. And the method here is that we use a bioinformatics software that is called Mixer, which Mixer will take your samples reads and align with the IMGT database's reads. So from the output of Mixer, we can just shows that the exact number of the mismatches in their VDJ genes. And we're just assuming that if we take European samples and then run Mixer, the European samples will have fewer mismatches, mismatches in their VDJ gene, substitution, insertion, and deletions compared to African and also Asian samples. Yeah, we're just trying to prove this. And this is just our basic workflow. We just take published data sets from SRA, and then just run the data sets through Mixer. And then from the output of Mixer, we just try to analyze the mismatches, which is the substitution, insertion, and deletions of their V and J genes for VDJ genes. Yeah, this is just give you a quick overview of the Mixer's output. You can know that Mixer shows that the exact number of there's a lot of numbers here. I just give you the most important part. The mutation parts, you can show that there's a lot of there's a lot of S stands for substitution, I stands for insertion, D stands for deletions. This is just a method. Let me just give it. Yeah. And we also try to examine the effective read lengths, which is the exact read length that our samples started to align to the IMG database's reads. So yeah, this is our method, and this just give you a brief figure about it. Yeah. And for our current analysis, we just take like 11 Asian samples, 26 European samples, and then 6 African samples to run mixer and just trying to compare the mismatches. And these are their SRA accession number here. From our preliminary results, and this figure just shows that the number of substitution on their V genes and J genes, we can really found out that the European ancestry have fewer mismatches compared to African and Asian sample on their V and J gene substitutions. Yeah, this figure, this just gives you a quick overview about others like insertion and deletions across like 3 ancestry groups. And we just basically, we just do a statistical test over it. We first like use box count transformation of the data and then try to conduct like t-test over it. And we do found that the V genes like in the V gene substitution and the J gene substitution, insert substitution and deletions are statistically significantly lower in the European ancestry samples comparing to the African samples. And for like the comparison of European and Asian samples, we can also show that the V genes in their substitution, V and J gene like substitution, insertion, deletion are statistically significantly lower in the European samples compared to the Asian samples. Yeah, this is just a conclusion of our studies. Just of our current result, the European ancestry are more completely representative of the IMG databases comparing to the African and Asian samples. And we're currently trying to expand our analysis over the entire like published TCR dataset on SRA with available ancestry information, trying to see if it is the case. And we just hope that by this result, we can just try to increase the diversity in like not only the immunogenetic database, but also increase the diversity in like all like field of omics studies. Yeah. Thank you all for your listening. And if you do you got any questions?
So can you go back to slide with the number of this one? So why is African so much better than European?
I think that's just like well, I'm still investigating it, but I think I'm still investigating it, but I hope that I'm not sure if we increase the sample size of African ancestry, will it still be the case since we're analyzing like only 6 African samples for now, but in the future we're not sure like how the result will be in the future, but we'll see. Yeah, it's an interesting point to investigate. Yeah, thank you for your questions.